All right, so how's everybody doing today? Everybody doing all right? Oops, my camera is shut. Okay, did you get a chance to read what's on the screen and help me answer? Has it been a crazy day? I see Courtney answered our question. Does anybody else have a question? I mean, an answer to the question on the board? What do you think? What technology could have helped us with this problem? Oh no, Irene. Just tell me, read through this little story here and then tell me what technology we have today that might have helped with this case. So Courtney says the comparison microscope to compare the chemical substance of the eggnog to the arsenic poisoning. Okay, that's possible. I think we can do it even more efficiently though. What other things do you think? Or what's the process in general that we use to help us figure out chemicals and poisons and all that good stuff? Okay, blood analysis. There you go, toxicology reports. Yeah, toxicology. And we use all these fancy machines these days, like gas chromatography and all these just huge fancy machines in the lab that can run through and do things that we could not have done in the 1800s. And so what another thing that somebody else pointed out is that maybe we could track financial records of the lady that bought the arsenic, because people back then used cash, but people these days mostly use debit and credit cards, so there might have been a paper trail. Okay, so good job. Yeah, we have our toxicology system really refined these days, and we are able to obviously test for things like arsenic. All right. So now I have an instant poll here. Let me see if I can get this to work. Okay, I think it came up on your screen. Help me answer this question. What were the names of the Spanish doctor and the chemist who found a way to detect arsenic? What do you think? All right, got some answers coming through. Got 7 people answering so far. And don't worry, I won't put your name on the board. I'm just going to put the chart up on the board. Some people haven't been doing their reading. You didn't remember from yesterday. It's okay though. I'd rather know that you don't know the answer. Can I get some more responses? We have 8 so far. Yeah, but be careful with that. Okay, so this is what you guys answered. Unfortunately, it's not correct, but that's okay because we can fix it. So a lot of you said Michael Gonzalez. Why does that look the way that it does? The correct answer is Matthew Orfila and James Marsh. Okay, so Matthew Orfila and James Marsh were the ones who figured this out. Yeah, yay for you if you got the right answer. What is Matthew Orfila considered? The father of what? Yeah, the father of toxicology. Good job, Courtney. So when we're talking about Orfila, we're talking about toxicology. He was one of the ones that helped us discover a way to detect arsenic. Awesome. Okay, so we're just going to cover some more of the history of forensics today. Last time we talked about some fingerprinting and how forensics gained traction. All the way up to the 1700s is when it really started picking up popularity, and Doyle coined the term, or didn't coin the term, but helped make popular the term detective. And then we reviewed over Frey versus Daubert. Does anybody know how to summarize what the Frey standard was? You have a quick summary of the Frey standard? Okay, that would be for the polygraph test. So that's what it was talking about, was it was trying to decide if the polygraph test was admissible. There you go. If it's accepted by the scientific community, it could be accepted as evidence. And is this the standard that most states go by these days? Yes or no? Is this the standard that most states follow? No, that's right. We go by Daubert. And what does Daubert say? What does the Daubert case say? Okay. I'm going to let Irene finish typing here too. Yes, exactly. So good job. The Daubert standard says that the judge is the gatekeeper of what is admissible into court as evidence. So for each different style of evidence, like for example, a polygraph test, the judge would determine whether or not that's an acceptable piece of evidence. And this is the one that we follow most days now. In most states now, not most days, all the days. Okay, so I'm going to copy this to the whiteboard, and it has worked in my past 2 classes, so I'm hoping it will work here. I'm going to unlock the whiteboard, and you should be able to draw lines and things to connect these terms to their definitions. So use one of these whiteboard tools and help me to connect the words to the definitions. I'm thinking maybe it's not working. Is it working? Yes, you can use the drawing tool, the paint tool, the line tool, whatever you want to do. Okay. And then if you see any ones that you don't agree with, you can change them. So we can have everybody answering these questions if you want to. What do we think? Anybody going to help them out? There's 2 that are wrong on the board. Are we all paying attention? Okay, good. Can we fix the ones on the board that are not right? We got one of them fixed. Oops, I did something wrong. Oh no, what have I done? Let's see if I can fix it. Undo, undo. Okay, fixed it. Okay. All right, I think we have all of them answered, but one of them is still wrong. Look over these and help me fix them real quick. What about this one, right? Oh, I can't click on it. Do we need some help? Okay, let's look at the answers. Oh, it was down at the bottom, Jeremiah. I don't know if I did something wrong. All right, so for the most part, we got this right. So nucleotides are the building blocks of nucleic acids. Deoxyribonucleic acid is the chemical name for the genetic material DNA. The base pairing rule is the predictable matching of pairs of DNA bases. That's the one that we didn't get right. Then genes are the linear DNA that codes for protein, and phenotype are the observable expression of two alleles or genes. So good job. You got most of those right except for that base pairing rule. So when we're talking about base pairs, we have four bases, adenine, thymine, cytosine, and guanine. And they pair always in this way. A goes with T, C goes with G, okay? And you remember that by saying apples in the tree, AT, and car in the garage, CG, okay? So always adenine pairs with thymine and cytosine pairs with guanine. So let's watch this little video kind of as an introduction to DNA, and then we'll come back together and talk about it. You can click right on the video to get it to work. All right, so you should be done with that video. Yeah. Didn't you also love his not his DNA, his his accent? He's like, you get your different suspects with your different samples. It's so British. Love it. Yeah, he's very enthusiastic. So what did you learn about DNA in this little video? How do we compare different DNA from different suspects? Different suspects in British. Yeah, it's different to everyone. And what do they do? Yeah, they have different markers after they run it through that gel electrophoresis. And I wrote here on the screen that scientists compared 13 different loci. So 13 different points on those DNA markers are what they compare in order to see if it's a match or not. Yes. Isn't that cool, Irene? It's called gel electrophoresis. I think that's just amazing. First of all, that people could figure that out. And then second of all, that's like, I can't even fathom that like everybody has different DNA. It's so cool. Maybe I'm just geeking out over here and nobody's actually that thrilled, but I think it's amazing. Okay, here is another video. We're going to talk about sometimes when scientists get these little teeny tiny samples of DNA. For example, if you've ever done an ancestry test, they send you these little swabs and you just like swab the inside of your cheek and you send that in, and that's how they run that test. First of all, they have to be very cautious to not contaminate the sample. So they have to make sure they wear masks and cover their hair and all that stuff so as to not contaminate the sample with their own. And then if it's a teeny tiny sample, they do something called a polymerase chain reaction. And that's how we amplify the small amount of DNA that we have to make it a bigger sample. It really works. It's amazing. So you can watch this video in on this page, and it's going to tell you about the PCR process. And just a warning, it has epic music. I guess all the videos that I use lately just do. But and then we'll talk about it after you watch this video. How cool is that? Are you guys geeking out as much as I am? I'm geeking out. That's an amazing process to me. Yeah, so cool. It's science. It's science, as they would say on Mythbusters. I just think it's amazing. So what are the things that they do? Can you tell me the steps of a polymerase chain reaction, or maybe you weren't paying close enough attention to remember any of the steps? What's the first thing that they have to do? It's twisted up in these coils, and so they take it and they do what? Anybody know? Anybody want to take a guess? If it's all twisted up, yes, then they have to denature it. They have to heat it up to get it to uncoil and straighten out, and then it separates into two different strands, okay? Yeah, exactly. And then what do? They put in some what? Polymerase. Okay, Ketsia. They put in polymerase, which is an enzyme, and they also put in some primers, and then what happens? This is the easy part. Yeah, okay. And what does that mean? It means that it is making what at that point? Yeah, exactly. It's putting together the pieces of the DNA, and then it gets ready to start all over again. And then we denature, and then we put in the, then we got the polymerase and the primers, and it copies, and then it starts all over again. All right, and when we do that, if you think about it, if you thought about exponents in math, you know, you start with, let's say 4, just for the sake of time, and 4 turns into 8, turns into 16, turns into 32, 64, 128, 356, something like that, 256, 512, and then all of a sudden you're at like over 1,000, right? Just in a matter of a few cycles. And at the end of that video, they said in 4 hours they can have over what, like 1 billion copies. So I just think it's fascinating that you can do that with just a little tiny sample of DNA. And then that's when we run that STR, which is that gel electrophoresis, to look at the DNA and the base pairs and make a match. And we can look and CODIS to see if we already have a match in the database. That will give us something to compare it to. So this is what the DNA fingerprinting would look like. These are those different markers. This is not necessarily what it would look like in real life, but just an example. So it starts with a collection of cells or body fluids or blood, and then we extract the DNA using heat and an enzyme to break open the cells and free the DNA, because it's in what part of the cell? What part of the cell do we find the DNA? Think back to biology. There you go, the nucleus. And then we purify that sample because we got to get rid of all the cellular debris that we just created and any other impurities. And then we determine if there's enough DNA, and then we magnify that using our PCR, and then study it using gel electrophoresis. Okay. And the probability that you would have those same 13 loci of somebody else is 48 out of 10 billion. So not likely to happen. It's good enough for us to compare those 13 loci, because pretty much you're not going to match anybody ever, okay? Only 48 out of 10 billion times. All right, one more video for you all. This is about blood spatter. Now look at this word. It's spatter, not splatter, okay? Spatter is the noun, splatter is the verb. So blood splatters and creates blood spatter. and the movements that it creates can tell a story. Okay, so let's watch this video just as an introduction to blood spatter, and then we'll talk about what you learned. Did you notice how she giggled a little bit in that video? Did anybody else notice that at that one point? After she did that cast off pattern, she was just like giggling a little bit. I thought that was funny. Yeah. So what did you learn about blood spatter? I don't know if it was cute or creepy or maybe both. What did we learn about blood spatter in this video? Okay, we have impact, cast off, transfer. Okay, most likely tells that the death or injury was caused by force. Depending on what the blood spatter looks like, right, Asher? It will tell us the type of force that was used to kill the person or to cause the injury. What was that transfer called once she moved her hand? It was transfer with what? It's that Dora character. She called it a swipe, right? It was a transfer with a move, so like swiper. That was my hint. Okay, so it's transfer with a swipe. So that's just our basic introduction into blood spatter. Obviously, we're not covering all the details right now, but just wanted you to get an idea of what is involved in studying blood spatter, since we're talking about types of evidence. Okay, so we're a little bit early getting done here, but I did want to save us some work time. So two things coming up. One, you have your history of forensics discussion, and then 2, you have your history of forensics quiz. Both of those are coming up soon. They're due on the 22nd and the 23rd. But before you do your quiz, you need to use your guided notes to study. So I've posted this PowerPoint, including the slides from yesterday, on Canvas, and I would highly recommend using those guided notes to go through that PowerPoint and answer your questions. And that will help you study for your quiz. Don't get into that quiz and freak out. I mean, go study first, okay? But if you don't feel like you're ready for that, you can either study now or you can work on your history of forensics discussion. That might be a safer bet. So I'm going to give us 10 minutes to work on these things. I'm going to set a timer and stop our recording here, and then we'll come back together in order to wrap up class.